So as I was talking about the interleukins, so these interleukins are quite much involved in the visceral organs cancer. So when there is an increase in the nitric oxide radicals or the reactive oxygen species, these interleukins 1 beta activations generally takes place. Or any factors like the uh, toxins from any organisms, that is again the damage associated molecular pattern. So these all leading to the activations of the nucleofactor kappa B, which in addition with the other factors like inflammasome factors, they actually increase in the inflammations. So overall, by the various factors and all that leading to the increase in the interleukin 6, interleukin 11, as well as the is a STAT, that is the signal transducer activator of the transcription. So these all things are generally being increased and leading to the inflammations, gastritis, which further progress into cancer. Move. Next. So the risk factors that are associated with the adenocarcinoma, the genetic susceptibility as well as the non-genetic factors. So the genetic factors I have already said, there is a single nucleotide polymorphism. Apart from that, the polymorphism or the changes in the long RNA or the microRNA. So these are all generally being seen, and this microRNA is of a topic of interest as because there are two types of microRNA are there. One which leading to the cancer, and another one is actually for the inhibitions of the cancer or for decrease the chances of the cancer. And the other non-genetic factors are there H. pylori infections. Apart from that, GERD, that is gastroesophageal reflux disease. Okay, move to the next slide. Over here, as I said, the H. pylori that consisting of the CAGA factors or the cytotoxins. So this CAGA leading to the activations of the protein kinase B or the ERK protein, which further activates the human homolog of double minute 2, which undergo phosphorylations and further is leading to the ubiquitinations of the P53, that is the guardian genome protein molecules, and that leading to the proteasomal degradations. On the other factors, it directly activates the nucleofactor kappa B, which leading to the inflammations or can cause the AID mutations, AID activations, which leading to the mutations of the P53 gene. So AID is the activations of the cysteine deaminase. So in other way, I can say that this all, whatever the markers of the molecules are there, these all molecules are the target for the gastric cancer. Next. So here similarly the influence factors. Now the influence factors over here, they are saying that there are certain receptors are there. On the left side you can see that is the receptor tyrosine kinase are there, vascular endothelial factor receptors are there, platelet derived growth factor receptors are there, fibroblast growth factor receptors are there. So these are the various receptors or the protein associated on the cells. They are actually the targets of the several drugs or several monoclonal antibodies. So these are if there is an increase in the expressions of these receptors or these proteins, then there will be the downstream hyperactivations that can lead to the cancer. And over here in the different phase, the different drugs are in the pipeline has been shown. They are target for the different growth factors. For example, Elotinic is generally being targeted for the receptor cell surface associated receptor tyrosine protein kinase. And there are certain molecules are there like telatinib, masitinib. So these are the certain molecules which are being approved by the FDA. Next slide. Okay, these are the certain tumor markers are there. So there are certain tumor markers, among them few tumor markers are the universal markers for all the different types of cancer. For example, CEA. CEA is a carcino embryogenic antigen. Other than we have the carbohydrate antigen 19-9. So and the other antigen 72-4. So these are the certain markers, are the classical markers which helps or aid in the detections of the cancer from the liquid biopsy, that is from the fluid, that may be the blood or maybe the serum or maybe the tumor fluid. So generally in that, these are the markers are generally seen or generally estimated from the biological fluid for detections of the cancer. Next. So as I said, the miRNA is one of the influencing, the markers is there. So here in this table, there are several miRNAs been shown, like miRNA 21, 22, 29C, 141. So miRNA is the microRNA, or they are the small interfering RNA, micro interfering RNA. So they are actually hardly 20 to 25 nucleotide sequence is there, and this can be used as a marker for the early detections. Now early detections is very important. If you are try to detect the disease at the early stage with certain symptoms in the patients, then that is very much needed of estimations of these early markers. Otherwise, if the case reaches to the stage 2, stage 3, then it will be very difficult to the patients to be survived for the longer time. At the early stage detections, if can be done, then the patients can be survived for the lifespan can be increased in the patient. Okay, move to the next. So here you can see the biomarkers. Apart from that, predictive biomarkers are there, which are being commonly used. High risk biomarkers in case of the high risk, and in case of the molecular subtypes are there. Treatments based biomarkers are there. Mostly they are tyrosine kinase receptors, as well as the apoptotic certain ligands are there, and the others like DNA methylations and certain apoptotic proteins or anti-apoptotic proteins can be detected as a markers. Next slide. So among that, one important marker is the PDL1 or the PDL. So generally two pathways used by the gastric cancer cell for the upregulations. PD1, that is the programmed cell death receptors ligands, and thus avoid immunity. The first one is normally involved in initiate the immunity response, and the upregulations of the PDL1 so can be caused by the activations of the oncogenic signals, either by AKT activations or the phosphatidylinositol 3 kinase activation. So these PDL1 expressions, if it can be decreased by blocking the protein kinase B or by blocking the phosphatidylinositol 3 kinase, then further it will be not able to, tumor cells will not able to interact with the T cells, and thus the proliferation of the cells can be stopped. Next slide. So as I was saying, there are certain PD1 and the PDL1 blockers are there, or the inhibitors are there, and some of the inhibitors like nivolumab, atezolizumab. So these are the monoclonal antibodies are there, which are being invented by the several companies and marketed by the several companies, and as they got approval long back in 2014, 16, and 17. And there are a lot of this PD1 inhibitors research is going on in the different sectors of the cancer institutions or the cancer research companies or the different research laboratories. Next. So this has been already covered, the recent therapeutics. So move to the next. Okay, so what will be the future research? Future research is the, there are a lot of genetic studies must be done. There are a lot of early detectors or the early markers must be detected, and the germline genetic studies must be done. So these are all the things to be done, and also the lifestyle must be changed, or certain environmental factors must be controlled, and there will be detections of the several genes or the markers should be done. Next slide. So in the future research, we can go with the certain things like, increase the persistence of the, prolong the half-life of the, and controlling the effect of the progressive release and the active drugs, conjugated polymer tags. We can either we can with the certain interleukins, we can add some adjuvant drugs, or we can increase the anti-inflammatory recombinant interleukins we can synthesize, or we can produce, which can decrease the chances of the gastric cancer. And also with the combinations with the toxins, or the combinations with the antibodies, these interleukins can be delivered for the treatment of the disease conditions. Next. What is the further prospects or the future prospects? Now, whatever the treatment we have, the chemotherapy treatment and all, we believe that this chemotherapy treatment and all can increase the survival time of the patients up to certain extent. But in addition, also we need a gene therapy or the genetic therapy, or if we can have some certain techniques and all that can leading to the targeted therapy towards only the cancer cells, or like using the nanoparticles and all. So that can be very much useful. Apart from that, if immunity can be developed against the cancer cells, so if the immunity can be developed by using certain immunotherapy or certain the drugs which are involved in the increase in the immunity, so that can be also be useful. Apart from that, we can also go with the apoptotic protein targets like PDL1, which is already been in the scenario. Okay, next. So operations generally, academic operations can be done, and the research experiments, we generally have a very good laboratory facilities and all. So over there, the research experiments and all can be carried out, and in collaborations, publications, and the patents, as we are in also in patents. So the searching of the better outcome of the research in collaborations can also be done, and with that also the laboratory corporations and the journal findings of the journals, or the suitable journals with the better quartile or the Q1 Q2, and also in combinations we can look for the international funds for the development of the both of our side. Next, these are the certain references. Next. Thank you for your attentions.
Okay, thank you, Professor Firdous.